Can you hear me okay? It's hard for me to know. And welcome to the people on Zoom. Oh, now I hear myself going through. Yes, I'm Nancy Reed. I'm here in an artificial capacity to introduce CANSI Ontario because the director, Lisa Strug, is away, and so is the deputy director, Rohan Alexander. I was the director of CANSI National when we launched the program of regional centers. So I'm very excited to see them all starting to take off now and have lives of their own. These are the members of CANSI Ontario. So the CANSI National is headquartered at Simon Fraser, and there are regional centers in Atlantic Canada, in Quebec, Ontario, and in the prairies. And these are ways to kind of keep the national flavor of CANSI alive while adapting to regional needs. And these are the university members of CANSI Ontario. There's this whole suite of programs that have been developed for CANSI Ontario. There's the Banting Discovery Awards, a training program in genetic epidemiology and statistical genetics. There's various data access grants. There's a postdoc program, 2 postdoc programs, and the various lectureship programs. And we have, during the year, we have regular seminars on Mondays called the ARIES Seminar, Applied Research and Education Statistics seminars. There's also the STAGE program. We have a software conference, and Rohan often runs a workshop on reproducibility. But the highlight of the year is the Distinguished Lecture Series in Statistical Sciences. It's sponsored jointly by CANSI National and CANSI Ontario. Our next stage speakers series lecture is in June. Paul Farrow will be visiting from Los Angeles, and then our applied research seminars starts up again in the fall, and Rob Cold will be here in early October, late October. That's it. Thank you very much for coming. It's my duty now to hand over the microphone to Martin Lisi, who will introduce the lecture today.
Thank you. Thanks, Nancy. My name is Martin Lisi, and it's my great pleasure to introduce the Distinguished Lecture Series in Statistical Science, as well as our speaker today. The DLSS was established in 2000 by Don Dawson, who was then the director of the Fields Institute, and our very own Nancy Reed. And since then, it has moved over to CANSI. It's a prestigious award consisting of a general lecture and one more specialized lecture delivered by an internationally prominent statistician or data scientist. Here's this year's nominating committee. I'd just like to thank my fellow members of the committee for this process. So it's the speaker goes through a rigorous selection process, which is mandated to evaluate merit through the multiple lenses comprising CANSI's vision for impactful research in statistical sciences today. So here's the photo of all the recipients of the DLSS so far. It was established in 2000. There are 22 speakers. And before you can identify which year didn't have one, I'm going to jump in to introduce this year's winner. Eric Joel Tchetgen Tchetgen was raised in Yaoundé, Cameroon until the age of 12, and then went to boarding school in France. While completing his Bachelor's of Science degree at Yale, which by the way was in electrical engineering and applied mathematics, he received a 1-year travel fellowship to go to Ghana. While he was in Ghana, he used his mathematical modeling to understand what factors are important to the adoption of new hybrid maize seeds. And it was while doing this work that he realized that he could use his math skills in order to promote public health. He came back to the US to complete his undergraduate degree. And after that, joined the Harvard School of Public Health as part of an internship where he was a statistical programmer. And the rest is history. Specifically, he obtained his PhD in biostatistics from Harvard University in 2006, after which he served on his faculty from 2008 to 2017. After that, in 2018, he was named the inaugural Luddy Family President's Distinguished Professor in the Department of Statistics at the Wharton School at the University of Pennsylvania. He has well over 200 papers in top statistical, epidemiological, and medical journals, having made numerous influential contributions to development and application of statistical methods for missing data, causal inference, semiparametric regression in social, genetic, and infectious disease epidemiology. A very long list of awards. I've highlighted a couple here. The Kenneth Rothman Epidemiology Prize, the Mirto Lefkopoulou Distinguished Lectureship, and most recently in 2022, the inaugural Rousseau Prize for Statistics, which comes with a $1 million cash prize and is presented by Their Majesty the King of Belgium for his work on causal inference with applications in medicine and public health. So without any further ado, I will turn it over to Professor Tchetgen Tchetgen.
Thank you so much for that warm introduction. Thank you to Kansi for this amazing opportunity to come and speak to you and present some of my work. It's been an amazing visit so far. Toronto, the weather is nice, and I've made a lot of new friends. I've seen old friends way back from my years in Boston, who our paths are again crossing. It was really pleasant surprises, and making a lot of new friends as well. And so, you know, we live in the age of machine learning, and so I had to give a talk where machine learning had to appear somewhere in the title. And if I asked ChatGPT to suggest a title, given my CV, this is probably what it would have come up with. So let me jump right in. It's not moving. Okay, I'll use the mouse. So here's a quick outline of my talk. So I had the good fortune of starting my academic career actually as a faculty in the Department of Epidemiology at Harvard, and eventually moved over to biostatistics. This was primarily because of strong suggestions by the students in epidemiology that maybe I was better suited teaching biostatistics students. And so I took their advice and I don't hold it against them. But I learned some great lessons from epidemiology, and that's often where I go back for inspiration for my work and my application. So I'm going to try to explain the role of motivate the talk by giving a framework for the role of causal inference in epidemiology. And there may be one or two formulas in the entire talk. And so hopefully this is going to be appreciated. The talk is going to be appreciated by a wide variety of audience with diverse interests who might be interested in statistics, in machine learning, causal inference, or public health. So I'll start off there. Then I will talk about a very strange combination of technologies that I've had the opportunity to work with. I refer to this as opportunities for holy union that brings together causal inference, semiparametric statistical theory, and machine learning. And if you understand at least one of these, you're in great shape. If you don't understand any of them, you're in even better shape. So the union is going to be about selective machine learning and the role of you thinking as in terms of there are a lot of options for machine learners out there. If your goal is specific to trying to evaluate the effect of an intervention, and you're worried about a high dimensional set of covariates, the question at hand is which technology should you use for your given application? And so that's what I'm referring to here as selective machine learning, and I'll describe the framework in which this came up in my work. Then I will move on to a slight shift gears a little bit and talk about actually an experience I had in industry. This was working with Amazon, and a tool that came out of that experience. And so this was grounded actually in very pragmatic, business-oriented question. And out of it actually came what I was very proud of our group, a tool called Credence, aptly named Credence, to make causal inference more credible to users in their business space. And then I will then move on to conformal prediction. Conformal prediction, if you haven't heard of it, is a fantastic development in statistics that promises amazing results. And so I will demonstrate an application of conformal prediction in my world. And then finally, this is all wonderful opportunities of machine learning and semiparametric theory and causal inference coming together. I really try to be upbeat in this talk, but I will try to close with a slight, maybe not warning, but also to be on the lookout for the possibility of this union turning unholy and potential risks. And I'll focus primarily on one prominent example in the literature recently called the Deconfounder, and maybe talk a little bit about that. That's just maybe 2 slides of slightly critical, and then the rest of the talk is supposed to be really upbeat and positive. And so I'll spend most of my time covering that, and then I'll close with some concluding remarks. And if there are any questions, I can take them there. So let me start with epidemiology. The central roles of epidemiology are twofold. One, which is the most common one that you hear about, which is a more or less descriptive role. And you can see this often in the newspaper. What is the distribution of disease frequency? We heard a lot of this about COVID, right? Incidence of COVID over the pandemic. We also hear for non-communicable diseases such as cardiovascular disease risk. Often one is interested in what is the cardiovascular disease risk among those individuals who exercise or don't exercise. This is purely descriptive, right? This is what we see, and we're just describing that what we see with those numbers. Causal inference is entirely different. Causal inference is trying to determine what are the causes that lead to those disease frequencies? Are they interventions that we can make to potentially change the frequency of those diseases? Does exercising reduce your risk of CVD? If possibly contrary to fact, you were not someone who exercised. That is a kind of factual question. And thus, it's a little bit in the realm of the metaphysical, because we imagine not what is, but what could be. And so that's essentially, I think of myself as essentially one who works in fiction. I write fictional papers about kind of factual worlds and potential impacts of intervention and the tools for that. And our role as causal inference people within the context of public health is to try to encode assumptions under which that fiction can become more and more close to reality. How can we actually learn from that language, that framework, about potential interventions that would be useful to our existing reality? And so causal inference is about, if I had to summarize it in one sentence, how to estimate the causal effect of an action on an outcome of interest. There's a long tradition of this in science. In fact, this goes to physics, often ask similar questions, chemistry, biology, a lot of the basic science. But a lot of those sciences have the advantage that it's possible to often do experiments. So experimental designs feature prominently in those disciplines. I'm more interested in, and I'm going to focus primarily in the social and health sciences, epidemiology being the primary example, but also economics, sociology, in which cases the opportunities for experiments are often limited. And so one has to resort to observational design. One prominent example that I encountered recently and is highly of public health interest is in fact in the context of COVID-19 vaccine. As we all experienced, early stage of the pandemic, companies were rushing to develop drugs, and they actually did use experiments to decide whether or not those drugs were efficacious. In this particular case, vaccines were the first round of therapies or preventive therapies, and those vaccines were shown to be efficacious through randomized trial. Pfizer were the first to run a randomized trial, then Moderna, and so on and so forth. And that was wonderful. That gave us amazing vaccines that improved our prospects for staying alive. The next question that arose was, with the changing conditions of the pandemic, the variants, the circulating variants continuously changing, which vaccine should I take given my profile? Should I be, given the circulating vaccine, should I be taking the Pfizer vaccine or the Moderna vaccine? Now, if you try to ask such a question using data, primarily you would like to do an experiment, but unfortunately those two vaccines are from two different companies. And they're unlikely to want to have a head-to-head comparison. In fact, there were no head-to-head comparisons. So this question effectively has to be answered using observational data. And in fact, that's what happened. People followed individuals. There were various designs that were taken, but they were all observational. And in fact, there was evidence that one vaccine was better than the other. And I'm not gonna say which one, depending on the variants that was circulating, okay? And so this is just to give you, to crystallize this idea that often in our recent history, in fact, we're faced with not having access to experiments, and we have to make imminent decisions that affect all of our lives. Now, going back to the role of causal inference in epidemiology at large, there are generally two notions of causation. The first one asks a question about the cause of an effect that you've observed. First, you observe an event, you observe an outcome, and subsequently you identify the causes or events that led to the observed outcome. The second one, type of notion of causation, is in fact the reverse, the effect of a cause. You want to assess the effect of a well-defined exposure or intervention you have in mind on an outcome you have in mind. Classical example would be, does smoking cause lung cancer? Does AZT is one of the very first HIV drug that was approved in the United States. And the question at the time was, does it actually prevent the advent of progressing from HIV infection to full-blown AIDS? And so you can imagine there are many such questions that one could pose. An example of the first kind, the cause of an observed effect, actually one of the most important examples, I think, in the history of epidemiology, goes back to the 80s when we observe an unusual high number of patients dying from a combination of syndromes, including a rare form of Kaposi skin cancer and pneumonia. And the primary scientific objective at the time was to identify the cause of that outbreak. Eventually, HIV was found to be the cause. Today, I'm going to focus on the second type of question, the effect of a well-specified cause, primarily because I say here, it's the most common question that's asked in biostatistics and epi, but truthfully, it's actually the easiest of the two questions. And that's because the first type of question typically requires complete or near complete scientific understanding, a lot more than the second type of question, though the second type of question does require some some understanding, scientific understanding to some extent. These types of questions, such as two, falls under the experimental paradigm, which is made explicit in the context of a randomized experiment, but as I've argued, is still a useful paradigm when experiments cannot be performed for either practical or ethical reasons. All right, so I've tried to make a case for the need for thinking, reasoning about causal inference. Now let me try to say it more formally. Why do we need a formal theories of causation? Well, there are actually very straightforward reasons. One, it makes explicit what we mean in plain English by causal effect, for instance. How do I translate that into something that we all agree on? That is, what is, to speak like a statistician, what is the estimate we seek? And we agree on that. It explains popular sayings such as association is not necessarily causation. Therefore, standard statistical regression or correlation measures may not be used to infer causation in general. And so that's often something that you probably have heard and caution when you're interpreting correlation, not to interpret it as causation. I think that's actually an important role of a theory of causation, but I actually think that more importantly, it gives conditions under which association is causation. When can I actually, when am I licensed to interpret association as causation? And therefore, standard statistical methods actually provide evidence for causation. And more important reasons to think about theories of causation is that it makes explicit assumptions needed for the identification of causal effects. And by that, I mean using data to infer about these fictional interventions. And allows for the derivation of new statistical methods when standard and familiar methods fail. And that's essentially how I've made a career. It's focusing on, in some situation, when can I think cleverly about ways to learn about causal inference when standard methods don't work? And you've probably heard of such techniques. Instrumental variable is one that's very celebrated. And in fact, that was awarded the most recent Nobel Prize in economics was for instrumental variable. That is a tool to reason about causal effects when you do not have a randomized experiment. You try to find a hidden experiment in your observational study. I'm not going to talk about instrumental variables today, but I do want to lay out the paradigm for causal inference. It's a very simple paradigm that is typically followed. One, defining causal quantities, typically done in terms of counterfactuals or potential outcomes. What would have been your risk of stroke if, contrary to fact, you had not taken your high blood pressure medication? Those types of questions. And then we'll compare it to what actually happened to you. And that's a notion of causal effect. Formulating for assumptions sufficient to identify, that is to learn from data, and quantities, and non-parametrically here, I mean without assuming functional forms, without assuming that the world is linear like in linear regression, just purely identifying from the data without any additional assumption about the data to identify causal quantities. And these are often, and don't worry about the meaning of these quantities, positivity, consistency, and unconfoundedness are types of such assumptions. Defining a mathematical model to deal with the curse of dimensionality. And this is an important concern in our era where we have access to high dimensional data. In the setting of medical records, you have available all medical records, all variables, all lab values that were ever measured on that given patient. This is potentially very high dimensional. By that I mean there are many such variables, often that scale with the sample size. How do we deal with these in settings where we want to use those data to learn about causal inference, which is a very different setting than to try to learn about prediction. And I'll come back to this in a few slides. All right, so that's the first part of this triad, causal inference. The second one is semiparametric theory. So let me try to convey the idea and the need for semiparametric theory and why I found it to be a useful tool in many settings. The idea is that in well-designed modern observational studies, one often has available to, in the data, a high dimensional covariance. Again, these are many features of units. Units will typically be, in my applications, individuals, patients, participants in studies, so on. And you would have variables such as age, sex, and social demographic variables, any other lab values that might be available in the medical context. And these are useful to address confounding concerns. So let me explain confounding with a couple of an example. Let me go back to a favorite topic for today, COVID-19. And this is actually referring to a recent paper in the New England Journal of Medicine. And that was evaluating the comparative effectiveness of mRNA COVID-19, the Pfizer vaccine versus the Moderna vaccine in the context of an observational study that was being conducted using the US veterans data. And the concern there is they want to do a head-to-head comparison between these two vaccines. And they're worried that individuals who are taking one vaccine may a priori be different from individuals taking another vaccine. For instance, if you a priori believe that Moderna is more effective, whether that's true or not, than Pfizer against the circulating variant, you may be, and you're receiving Moderna, you may also be more willing to go outside and party like it's 1984. Therefore, higher level of exposures to COVID-19. So that even if the two vaccines are the same efficacy or effectiveness, those systematic differences in the person's behavior, taking one vaccine versus the other, would lead to differences between the effectiveness that's being measured that are not due to differences in the vaccine, but rather due to changes in behavior. And that's a form of confounding mechanism. So to address this concern, these authors try to collect as many covariates needed to try to control for those differences, a priori differences between folks who took one vaccine versus the other. And as I mentioned, age, sex, race, ethnicity, urban residence. And these are variables typically that have been found to be predictive, not necessarily in a causal sense, but have been shown to be associated with higher or low rates of COVID vaccine effectiveness. Sorry, COVID incidence, right? So sex and age certainly was a risk factor for COVID because individuals might be more immune compromised or living in a situation where they're more exposed, like retirement homes. Sex, race, ethnicity, urban residence were all found to be associated with higher rates of COVID, not necessarily in a causal sense that they cause higher rates of COVID, but rather because they tended to be associated with different living conditions that would affect your level of exposure. And so this is the kind of confounding mechanism, trying to measure such covariates and adjust for them, we'll try to adjust for such a priori differences and the bias associated with them. Another important source of confounding is actually healthcare seeking behavior. If I'm a person who has a healthcare seeking behavior that will lead me to be more, to higher proclivity to get vaccinated, but also to lower proclivity to go and party like it's 1984. I'm old enough to go back to 1984. Okay, and then you would worry that that would again be a source of confounding, potential source of confounding, because that would mean you will be less likely to get the vaccine and you'll be more likely to be vaccinated. And this can go in either direction. And healthcare seeking behavior is actually really hard to measure, and so people will typically use proxies, such as how many recent vaccines, flu vaccines did you take, how often do you come to the hospital in the past for emergency reasons or non-emergency reasons, like you have an ingrown nail or a toenail or something of the sort. And even if willing to assume that we've measured all relevant covariates, that is unconfoundedness, given measured factors, How should these factors be accounted for explicitly? And so this is where, in fact, there's been a long-standing debate in the literature. There have been two strategies that have emerged over the years. One strategy is to model the disease occurrence as a function of the treatment and the risk factors, and that's typically done in a regression fashion, such as or logistic regression, or to use so-called propensity score modeling strategies. And these are incredibly clever methods that recognize that, in fact, all you need to learn is the association between these factors, these risk factors, and the treatment, and not look at the outcome directly and account for confounding through these tools. And these are known as propensity score modeling. These, if you're familiar with likelihood theory, are modeling different parts of the likelihood. And both, on the correct models, will identify the same quantity. And so you have to use statistical methods to decide how you're going to model those two parts, but you essentially have two independent ways of getting to the same quantity by building different models. And there was a long-standing debate of which approach is superior. And by superiority here is to note that epidemiologists are incredibly risk averse, and what they want to avoid is bias, not necessarily efficiency. Bias above all, because they do not want to make a statement that may be invalid. So this long-standing debate turned out to, of which is superior, was actually resolved by modern semiparametric theory. This is one of the tools, one of the amazing results that this theory has delivered. And it's actually shown that, as you would imagine or you would hope, you can do both methods and combine them in a very careful manner using the theory. And this gives rise to something that's been known as double robustness. So again, I have two methods for estimating one quantity. And these two methods can be modeled independently and combined in a certain way. And double robustness here refers to the fact that if either one is correct, without me knowing a priori which one is correct, I'll recover the right answer. And it also implies that, in principle, doing both can be substantially better than each separately in terms of efficiency. So this is a wonderful idea. Does it work in practice? All right, so I have to describe, there's a lot going on here. I have two quantities, alpha and eta. These are the two models that I said that I need to model. And I can identify some so-called causal effect by modeling either quantities. And I want to use standard parametric models, logistic regression models, linear regression models, the kind of familiar regression models that you can use using standard software by pressing a button and letting it run. No special coding required. And what I've done here is a simulation study where I have 4 scenarios. And each scenario tells me which model is correct and which is wrong. And I have 3 kinds of estimators. One that requires propensity score to be correct. One that requires the disease risk method to be correct. And one that is doubly robust and works if either one is correct. So in this first scenario, both are correct. I have bias, variance, and coverage. And the coverage here refers to if I want to construct 95% confidence interval, it better be the case that if I run my simulation 100 times and construct confidence interval, the confidence interval covers the truth approximately 95% of the time. So the key number here is 95%, 0.95. And I want to check that the coverage is close to 0.95 in my simulation study. And you can see that when everything is correct, the well is beautiful. All methods have a small bias. Bias we want it to be small. They have relatively same efficiency, and they all work pretty well in terms of coverage. In fact, you could focus on coverage here because it ultimately will tell you whether or not you will announce a valid result when there is not one above your nominal error rate. The interesting scenarios are when one of them is correct and the other one is wrong. So for instance, when alpha refers to the propensity score model, when alpha is correct, but the outcome regression is wrong, you can see that the bias blows up for the outcome regression model or the disease risk method. But the propensity score still remains unbiased and has the same coverage. In fact, it's exactly the same estimator. And what's interesting about this scenario is that the doubly robust method essentially agrees with the propensity score method. It adapts to the propensity score method results, whereas the disease risk model gets it wrong, and vice versa. When the propensity score model is incorrect and the outcome and the disease risk model is correct, you get the symmetric result. And what's nice again is that the W robust essentially adapts to which one is correct and gives you the right coverage. And so this is the promise of double robustness. But to be completely honest, I also have to present what happens when these are, after all, parametric model, when both methods are wrong. When both methods are wrong, all bets are off. The coverage is lousy. It's nowhere close to 95%. They both have bias, and so does the W robust method. So on the one hand, these simulations confirm that double robustness theory works in theory and in practice, at least in simulation settings where I know the truth, in the sense that it delivers valid causal inference if at least one of the two strategies can recover valid inferences without a priori knowing which model might be incorrect. And this has been an incredible success story in statistics. But I think there's another side to this, and I contributed to some of this literature. And I eventually came to the realization that this last scenario is effectively the most important one, because it exposed a major vulnerability of this idea of double robustness in a setting where you're using parametric models. As you've probably heard the adage that all models are wrong in practice, and some might be more useful than others. But in this context, all models being wrong can actually be worse than one model being wrong for the W robust estimator. And so we identify this as a double robustness paradox, that we have this beautiful method that came out of this beautiful theory. However, the W robust estimator may actually have much larger bias than non-W robust estimators in parametric settings. Acknowledging this paradox is actually really painful for me. I've spent my dissertation years working on this. And it validates any claim of superior validity made on the basis of double robustness in parametric case. And so this initial effort of Semiparametric to address this difficult problem is not a complete success story. So I'm now going to move to use the machine learning part of the talk. Machine learning might come to the rescue. Why? Because machine learning, the whole idea is to move away from parametric models and to try to use things that are very flexible, that adapt to the complexities and the idiosyncrasies of your data. So modern machine learning has given rise to several highly flexible methods for constructing predictive models in high dimensional settings: random forest, gradient boosting regression trees, lasso, LARS, elastic net, deep learning. These are, I mean, these names are incredibly evocative if you're familiar with some of this literature, and you can almost see what they tried to do by the name that they were given. Incredibly well-named methods. And there are a lot of those methods out there, and they all perform very well in some cases. But to my knowledge, no such method works well in all settings. And so the question is, can we recycle these methods to model the disease risk model and the propensity score, as these two models are inherently prediction models? And again, just to remind you the context, I'm interested in learning about the causal effect of COVID vaccine. So that's one number. What is the percent reduction in my risk for COVID that is afforded to me by using one vaccine versus the other? Okay. So it's just one number, but to do so with observational data, I must account for differences between people who receive one vaccine versus the other. And these differences are captured by features: age, sex, healthcare seeking behavior, so on and so forth. And I have a large number of such factors. And the question is, as a statistician, how should I model them? Double robustness helped a little bit, but it relies on making very strong assumptions about the models. Machine learning promises to be more flexible. The major challenge is that there are many such methods. They're hard to tune, That means, how do I find a machine learning that works really well for the data at hand, but also generalizes? And so the major challenge here is how to incorporate machine learning to fit these nuisance models, these regression models, flexibly, without compromising my ability to learn about causal effects. And there are two solutions that dominate the literature. There's one method called target maximum likelihood estimation. This is all lingo, and then double-debiased machine learning. I have never seen anything being picked up as fast as double-debiased machine learning by industry. In industrial context, often people are interested in interventions. And it's remarkable the speed with which this particular technique has been picked up, even while the paper was not yet published, was just up. And so these are methods that are being picked up really quickly. These ML approaches provide a principled framework for incorporating highly adaptive machine learning methods to account for high dimensional covariates. However, they are tuned to minimizing their prediction loss, not necessarily to balance bias and variance. Specifically, while machine learning method resolve this double robustness paradox because it frees you from having to use parametric regressions. You have now used these fancy tools. And therefore, it ensures that double robustness estimation bias is dominated by that of non-double robust estimators. That is, the double robust estimator is almost guaranteed now to have smaller bias. It doesn't guarantee that the bias is sufficiently small to learn accurately about the causal effects, which is again our end goal. All right, so let me just skip. I think I'm running a little behind, so I'm going to skip a few slides. I apologize. So what I want to describe is selective ML, and I want to do this by demonstrating its ability. So we have a recent methodology that essentially says, again, similar to double robustness, well, if I have all these techniques, and I've listed quite a few here, and don't worry about they're just different machine learning techniques that people in computer science and statistics in machine learning have developed over the years. And I want to know for a given data set which to use. We have a methodology which is called selective ML. And what I'm showing here is the ratio of mean squared errors in a simulation where we generated highly complex, high dimensional data, and we have various sample sizes. And the ratio here is comparing the mean squared error. The mean squared error is the amount of error that you're making in estimating your causal effect of interest relative to our method. So 3.3 here is essentially the mean squared error of this particular method relative to our mean squared error. So any number less than 1 does better than our method. Any number greater than 1 does worse than our method. And effectively, the bottom line is in these settings, in these experiments, all methods do worse. And essentially what our method is trying to do is going to use double robustness to define a notion of stability of estimators. And it will use that to select a particular machine learner. And this translates into a better practical performance. Selective ML is a promising advance. However, it does not completely solve the problem at hand. Mainly, for the data set we wish to analyze, which learner should we use? How can results be validated without knowing ground truth? Now, there's a big distinction between causal inference and prediction problems in the sense that in prediction problems, the strategy is I want to learn about what is the risk of this person developing cardiovascular disease in the future based on their data. So that's a prediction. You're trying to forecast a prediction. And what you would do is you would hold out a sample where you observe the outcome. You will use your training sample to train your learner, and then you will look at how the prediction does on the whole of that sample. The beauty of that framework is that you have the label, and you can do that exercise and you can tune your learner. Causal inference does not afford that luxury, because for causal inference, we're not interested in a prediction. We actually want to know about something that hasn't happened. We want to learn about, for a person who was treated and we observe the outcome, what would their outcome be had they not been treated? There is no holdout sample because for every single person, we only observe one of the outcomes, whether they're treated or they're not treated, and we want to learn about the other one. And therefore, tuning learners in such a problem is infinitely harder. And so the question that we were posed to, this is, I think I mentioned earlier, this was done in the context of consulting for Amazon. They asked the following question: every single researcher in causal inference have their methods that they like. And again, there's this overarching theme that is, for a given data set, which method should a person use? So I have a given data set and I want to know which of these 30,000 methods that are in the literature should I use for this data set? None of them dominates. So I think I have to skip a little bit ahead. I see they're setting up for refreshments at the back. What I want to say is the way we thought about this problem is ideally you would want what we call a bespoke simulation framework. And for that, we use deep learning technology known as variational autoencoders. And we develop a tool called Credence to validate causal inference. And I'm just going to say it very abstractly what the tool does. So this is in the context, think of you're now working at Amazon and you have data, access to all the data that Amazon has collected over the years in the particular slice of their business. And so we're going to learn this, we're going to train this machine learner over all of these data sets. This is not a small exercise. So, it's not as big as training it on the entire internet, but it's a lot of data. It takes sophisticated programming and careful tuning. And so what variational autoencoders do, so you have a family of data sets, you train them, and the encoding part translates each data set into part of a latent space, low dimensional latent space. And then the decoding part tries to reconstruct the original data set in your family of data sets. Again, encoding part takes your family of data sets, maps it to an underlying low dimensional space. And then there's another part of it, these are two neural networks, and then decodes it and maps it to a space in your family of data sets. Why are we doing this? Well, today, a scientist I'm working with comes up with a new data set and says, I want to know what method I should use for this data set I have at hand. I say, aha, your new data set is probably not unlike the history of data sets in Amazon. So what we're going to do is we're going to take your new data set, we're going to run it through the encoder, and it's going to map it to some place in the latent space, and we'll sample from that space locally in that latent space to construct simulated data that look exactly like yours, stochastically, but for which we have access to ground truth. And so these are what we call bespoke simulation studies. They allow you to generate simulation studies where you control ground truth in data that are anchored at your observed sample so that you may use those simulations to study various methods and rank the methods. So it's not telling you that you're recovering the truth, it's telling you for that data set, which method should you use. So just very briefly, there's a lot going on here. I'm just gonna focus on one picture and then move on. So I'll focus on, we did two kinds of evaluation, one based on a large simulation study where we generated a family of data sets, we trained Credence. So in this, the red line is the truth. The various colors are various estimators. They are all listed here. They are all published in the literature. There are a lot of them. They're all trying to estimate the red line. And so this is a simulation that's done using having access to the underlying data training mechanism. So this is an oracle would do. So what Credence will try to do is what you see right here. These are for two different mechanisms. So let's just focus on the left one for now. And this is what Credence will tell you. And notice something that in this particular evaluation, if you were an oracle that had access to the truth, you would say that this method in red is probably not one that you should use for this data set because it's far from the red line, the truth. But all the other methods perform pretty well, except for this blue method and the red method. And Credence under various conditions, invariably tells you the same message. Look out for that red method. Again, Credence doesn't have access to the underlying data mechanism. It uses your family of data sets and samples from the local latent space, local area of the latent space where your data set maps to. And so this was presented at ICML back last year as a technique. We also used it in two real data sets. And what I'm showing here is sort of just a correlation matrix. The color here is the elements of the matrix, the rows and the columns are variables. And so the color pattern is just telling you in the real data set, what is the correlation between the dependence between variables. And we wanted to see whether that's reproduced by the generated data in Credence. And the idea is to compare these two, and you see that the dependence is quite recovered using this tool. All right, I have about 5 minutes left. Is that right? It's until 4:30? Yes. Yeah, so let me wrap it up and leave some time for questions. I think I went a little over. Let me just jump ahead a little bit. So I do want to close with just this warning that I talked about some advantages of using these techniques and combining using leveraging machine learning to get sound results. I do wanted to give a warning that there are some areas, there's some principles of causal inference and statistical learning that should be preserved. Study design is important, is really important. Thinking about how you identify your models is incredibly important. Causal inference is hard. Ultimately relies upon untestable assumptions that cannot be ruled out or confirmed strictly on empirical basis. So in closing, I just wanted to mention this one example in literature. It's called the Deconfounder. And so beware of methods that claim that they can learn things without explicitly relying on assumptions that they cannot test. That's the trick. If any method says, I will give you the truth, regardless of what it is, and I'm not going to state what assumptions I'm making, you have to be making some assumption that's implicit or explicit. And so the Deconfounder is one such example. This paper appeared it's remarkable. So this paper is trying to learn about confounding, the main source of bias that I described earlier in the context of COVID-19. And it's claiming that it can learn about whether or not you're missing important confounders just by looking at the observed data. By definition, that should be impossible. How can I learn about things that are not observed? By looking at what I have observed without making incredibly strong assumptions. And so the Deconfounder by Wang and Lai has been picked up really, really quickly. It claims in the multiple treatment setting to learn about and correct for any residual confounding by repurposing the joint treatments to construct a confounding control variate using machine learning tools. In other words, all of the relevant information about selection into treatment that pertain to confounding can be completely recovered by the realized treatment. To be true, such claims must ultimately rely on hidden assumptions or a degenerate model of state of nature. Otherwise, they cannot belong to what Judea Pearl refers to as the second rung in the ladder of causation. And the Book of Why is a wonderful introduction textbook to causal inference for anyone interested in learning more. It's a popular book for that. And there are many papers that have talked at length about the issues with such techniques and warnings about how terribly wrong you can go, particularly in the context of public health. And you can be responsible for a disaster potentially by claiming such findings when it is clear that they are impossible. All right, I'm going to stop there so we have time for questions. I apologize for going a little over. I do want to acknowledge the NIH that has funded my research over the years. I also want to acknowledge my colleagues that have contributed tremendously to the research that I managed to describe here today. And I'll stop here and take any questions. Thank you so much.
Okay. Thank you so much, Professor, for this wonderful talk. So I'll just start by taking a couple of questions from the Zoom, since they won't be able to join us later, and we'll do a couple of questions, and if you would like to ask more, Dr. Tchetgen is still here, and we'll have some refreshments. You can ask him that. So I'll start with a first question from the Zoom audience. Is there any specific role of epidemiology to prevent Alzheimer's disease and dementia in a global aging population?
Yeah, that's a really great question. I think there is a growing interest in, obviously, Alzheimer's and related disorders are increasingly becoming a problem, not necessarily a new problem. We're just better at diagnosing it, and also people live longer these days. And so I think the science for prevention, I should first start by saying I'm not a medical doctor and I'm not an Alzheimer's researcher. I do have a grant from the National Institute of Aging, and I can talk to specific statistical problems that grant tried to address in the context of Alzheimer's. Part of the difficulty with such conditions is that invariably you have to use aging populations. And you're looking at, for instance, the impact for, when we're talking about prevention, for instance, the specific question we looked at was the causal effect of smoking when you're young on cognitive decline as you age. And the challenge there is the outcome is cognitive decline, which you have to observe individuals and measure their cognitive using instruments. And the challenge is that this is in an aging population. And so a lot of individuals die while you're following them up. So eventually all you're learning about is potentially the association between smoking behavior among individuals who happen to have survived long enough to develop Alzheimer's. That's a highly selected group of individuals. And what you're learning may be completely entangled with other risk factors for dying, which we know smoking is. And so there have been a lot of contradictory findings about preventive methods for Alzheimer's disease, including genetic predispositions that seem to, depending on who analyzes the data, seem to go in opposite directions. And I think that's mainly partly due, I should say, to this phenomenon of survival bias. Because you're studying a condition that's intrinsically going to be susceptible to such censoring by death, essentially.
Thank you. And there was a question from the audience here.
Hi, yeah, thanks for the great talk. Actually, the thing that you said was most kind of evocative to me was at the very beginning, framing causal inference as kind of approaching metaphysics, which I think is actually like precisely correct. And I was curious to hear if you had any thoughts on differences in the kind of metaphysical underpinnings of causal inference versus machine learning. Like your discussion of Deconfounder kind of touched on this, but I'm a machine learning researcher, if that helps contextualize the question. We often operate with this kind of like crude Bayesianism of just like more data is more knowledge. And that's really what we're being fed with the kind of public discourse on GPT right now. Yeah. So I just wanted to give you an opportunity to kind of any sort of thoughts that brings up for you. Yeah.
Wow. All right. You're calling me on some things that I said. So yeah. So to be clear, I mean, I think the overwhelming, hopefully the overwhelming message of my talk is that combining these techniques and these disciplines is a good thing. But my hope is that we can do so by respecting certain principles from each of these disciplines. So let's talk about causality and machine learning. I think part of the issue with combining causality and machine learning is that causality is intrinsically a missing data problem. Right. I have a person who received vaccine, the Moderna vaccine, and I know their outcome at the end of follow-up, whether or not they got COVID. And I want to know whether or not they would have gotten COVID had they not taken the COVID vaccine, the Moderna vaccine, they're taking Pfizer or nothing. And I'm doing so with non-experimental data. So this is a problem that cannot be solved by any kind of statistical method without making an assumption that cannot be tested. Because whatever assumption you're going to make is going to link your observed data to things that you have not observed, right? And so causal inference goes to length, and it's often described as being incredibly pessimistic. And I often get the criticism that causal inference makes the assumption of causal inference are too strong. And that's because the statement made by causal inference is strong. The only reason, and the main reason they're perceived that way, I think, is because it tries to be, the discipline as a whole tries to be upfront about the limitation of what we're learning. Now, I don't want to mischaracterize machine learning because I'm not a machine learning person. I picked on one particular paper, and that's not representative of the discipline. I mean, I've learned tremendously from machine learning. The one word of caution that I have is conflating what's put in a prior, particularly in Bayesian, what's encoded in a prior, conflating that with actually learning things that cannot be learned from data. And so if you make assumptions in a prior that are not identified from the data, they're going to show up in your posterior. You're just putting what you want to learn, what you're putting the answer in. You know what I mean? And so the only point I'm trying to make is, and not to drag this on too long, is careful consideration for some of these principles, I think, are warranted when adopting these new tools. I do want to say something positive, and I think we talked about this at lunch, which is I do a lot of work on negative controls as a tool for detecting biases in observational studies. So these are outcomes that you know cannot be causally affected by your exposure of interest. So for instance, in the COVID vaccine setting, flu outcomes are a good outcome because you know the vaccines cannot causally impact flu. And so by virtue of that, if you find in your analysis that the vaccine is protective for flu, that's indicative of confounding bias. There was a paper recently that came out of Microsoft, and I don't mean to advertise Microsoft here, but I was mind blown by this paper because it got me conflated, which used ChatGPT to essentially answer your question, what is the role? Because people say ChatGPT is going to, the Bayesian flaw is it can't reason about causality. And they took that on and they said, what is the role of ChatGPT in asking about causal statement? And one of the prompts was exactly what I said. I want to study our COVID vaccine. Can you tell me about what good negative controls to use? Because I'm worried about this confounding bias. And I looked at the list of things that ChatGPT spat out, and half of them I hadn't thought about. Then I looked at the ones I hadn't thought about, and half of them were complete garbage. They were not relevant for the source of bias. They satisfy one of the many assumptions that negative control variables have to satisfy. And the other half were completely new to me. And so I learned something. And so I don't want to throw away a whole amazing discipline that's going to bring us new tools. It's how we use them. It took iterations for me to say, okay, this was a useful exercise, is what I'll emphasize on. Sorry, that was a long answer. It's a great question.
I'll take one more from the audience, from the Zoom audience. I'll come back to the real. Okay, the next Zoom audience question is Credence generating synthetic data from the real data?
So the question is Credence generating synthetic data from the new data? So you can think of it that way. Credence is identifying the part of the latent space you trained in this library of data sets where your data set sits. It's a low dimensional space, and it uses neural nets to then recover high dimensional data that looks like your data. So if you run a bunch of statistical tests, you wouldn't be able to distinguish it from your data from the perspective of those statistical tests, but for it to be useful, it doesn't spit back your data. Otherwise, there's no variability. So you can think of it as really a deep learning version of the bootstrap, something that we're familiar with, but one that's highly effective. And so that an anchor can pick up on idiosyncrasies of your underlying data set that other methodologies might not pick up on.
Thank you. And then there was another question from the in-person audience, please.
I actually also wanted to ask about Credence a bit. And particularly, I wanted to point in a direction that you said you weren't going to talk about, but instrumental variables was sort of what I was wondering about. You've talked about this a lot in terms of the assumptions around confounding, but there's a different set of assumptions, obviously, for instrumental variables. Is Credence something that can be applied to look at those issues as well as and the latent space that would be with instrumental variables.
Yeah, thank you for that question. So I'm glad you asked that question because I think I rushed a little bit through it and I wasn't clear. So what Credence is good for is giving you something like this. If I have a data set and it turns out that if I were an oracle and I knew how the data set was generated, I could rank various methods to target a particular estimate, like this could include IV methods. I didn't include them. Credence will generate data based on one realization of your data set that look and generate a picture like this, and that's this picture. So Credence, the only thing that Credence does, it generates data in a particular fashion so that you always know what ground truth is, but the data looks exactly like the data that you observed. And so you can then rank. So this is an oracle that knew how the data was generated. This is Credence that learned how the data was generated from one sample, and then run a simulation and compare the various methods. So if you want to then include instrumental variable methods to this bucket, you can do so. What it's not going to tell you, all of these could be wrong. It could be centered around the wrong value. Why? Notice that I didn't have time to go into this, but I had different scenarios here. And I'm anchoring Credence at various causal truth. The causal ground truth is unknown to me. So I do have to go through this exercise of imagining, I wonder how the methods would perform under these conditions. I wonder how the methods would perform under this condition. The only thing that Credence does, it's essentially a fancy data training simulator. But it's a really good one in terms of mimicking the data at hand. And the reason that was important to the users here was because they wanted to answer the question, for the data that I see, which method should I use? And so it gives you these rankings, and then we just say, then it's a decision problem. You have to weigh your utility function or see what you care most about and then decide. So it's not an answer to the causal question. It's an answer to which method should I use for the spectral data set under these conditions. I hope that clarifies.
This is a tough one. I don't know how to say no to you, Jerry. Please go ahead.
Thank you.
You focused on average causal effects. Of course, that has limited applicability for somebody who wants to, for an individual person, in a sense. But I wonder if you could comment on the use of the machine learning models for conditional effects. Let's say you've got in an exposure or a treatment, and then just maybe a small number of covariates that you'd like to adjust for.
Thank you. And thank you for asking that question because it allows me to cover something that I couldn't cover during my talk. And so I'm going to go back to this, and I'm going to maybe spend 2 minutes, if I can, on explaining what's going on here. So this is using this framework of conformal prediction. Conformal prediction is an amazing set of tools that people have recently developed to do prediction at an individual level with marginal coverage guarantee. So let me just go back for a second. So what they want to know is, I have a data set, and you're going to assume that they're IID. So the data sets are coming from a same distribution, all of them, and they are N observations, and they're going to use a machine learner to get a predictive set to say, now, if I get an N+1 observation with a particular value of X, I want to know what is the set of potential values of Y. Okay. This is a prediction version of what you're asking about causal effects. You can ask the same question. And what the really neat trick that they did is they didn't ask about coverage being absolutely correct for every single person. What do I mean by coverage? It means the error by which you're misallocating the predictive Y to an interval. They want to control it, but they're not going to control it at an individual level. They're going to control it on the average. And so this is the criteria they use. The probability that your set contains a true value for these new individuals you see in the future, given their X's. That probability is marginal over both Y and X. It's not conditional. And they can show that in finite sample, they have a it's well calibrated. You give them an error rate of 0.05, you'll say, on average, 95% of the time, I'll get the right guess. So these tools are amazing. Now, the problem I'm interested in, the one you're asking about, is a situation where you have a similar problem, but you have what's called a covariate shift. That's another name for confounding in machine learning. And so the way it manifests itself is like this. This is the question you were asking. I want to know about the individual of these are potential outcomes on the treatment, on the control. I want to know about a set of values given a person's covariates. What is the predictive set for that, for those causal effect, individual level causal effect. And I want to control the error rate marginally. So I want to calibrate it very well. And so let me just go to tell you how this works. So we developed techniques to extend it to this setting. And just to show what this is showing is, on the left-hand side is the average coverage over simulations. And the nominal level is right here. And so on average is smack on. And the length of the interval is given right here. We actually rolled this out in an application. And very briefly, the application is a prediction application where we had, this is in Botswana, we had HIV incidence and covariates in particular regions of Botswana, and wanted to learn about incidence in a different region that looks very different in terms of the covariate distribution. So this is back again to this covariate shift problem. And we did it in a setting where we actually observed the labels in the new population, so we could validate our coverage. And what you see here is the empirical coverage of the technique. And these are different tools that we developed, the first two. And this is what standard conformal prediction. So standard conformal prediction that doesn't acknowledge the covariate shift doesn't have the nominal 95% coverage, but these techniques have the correct coverage. So this is the nice thing about these methods. You can guarantee the coverage of your prediction set. What's the trade-off? The intervals are wider. So for instance, to make this concrete, in the HIV example, we could do you're predicting sets. So you're either HIV or you're positive, you're not. It's a binary outcome. So these sets either contain one, that person giving their covariates is going to be HIV positive and did it 3 years down the road. So that's how much follow-up we had. 0, that person will not be giving their covariate, or 0 and 1. Guess what? None of the prediction sets included the singleton 1. A lot of them included 0 and 1. Sorry, quite a few included 0 and 1, and a lot of them included just 0. That's an incredibly useful tool because we don't want to claim that we can predict who's going to get HIV. We want to claim that we can do a better job at predicting who is not at risk for HIV, so we can focus on individuals who might be at risk for HIV. And so the fact that the confidence in the prediction intervals might be large is actually can be informative in that context, where then you worry about the covariate features of individuals who are more likely to be, who are not, who are at risk, potentially at risk for HIV. So that was again a long answer to your question, but so that was a good question. Thank you.
Thank you very much. That's all the time we have for questions. So just if you could please join me in thanking the speaker again before we go through refreshments. Thank you. Wow. Thank you. Yes, thank you.
Okay.